Kayli Bolton. I am a sophomore and biology major at the University of Kentucky, and today I will be presenting abnormal accumulation of glycogen and altered glycosylation in brain tumors. Imagine the University of Kentucky's rub arena filled completely to its capacity with people. This arena can hold 23,500 people, and yet to visualize the number of individuals expected to receive a brain tumor diagnosis this year alone would take nearly 4 rep arenas. It is estimated that 87,240 people will be diagnosed with a brain tumor in 2020. Nearly 1/5 of the individuals who receive a brain cancer diagnosis are expected to die fighting that cancer. And those who do live are at a higher risk of living with impaired cognition and physical health. The composition of brain tumors is both diverse and complex, and these qualities cause them to be difficult to diagnose and treat effectively. 2 characteristics have been connected to the progression of brain cancer: abnormal accumulation of glycogen and altered glycosylation. Glycogen is the main storage form of glucose, and as such, acts as an energy currency for the body. Glycosylation is a modification in which sugars called glycans are attached to biomolecules to modulate their structure and function. With these characteristics in mind, we conducted a study analyzing the molecular profile of 106 human samples whose initial brain cancer grade level varied from 1 to 4. We first conducted a tissue microarray sectioning of these samples to prepare them for tests. Then, the first technique we utilized was immunohistochemistry, or IHC as I'll refer to it throughout the duration of this presentation, coupled with halo imaging software. IHC is the traditional method whereby tumors are scored in the pathology lab, and is a technique to identify biomarkers taking advantage of the relationship between antibodies and antigens. The antibody that we used is against glycogen. The second technique we then utilized was mass spectrometry imaging, or MSI as I'll refer to it throughout the rest of this presentation. MSI is a novel methodology that has recently been used to analyze the sugar composition in lung cancer, but has not yet been applied to brain tumors. MSI is a powerful tool for the visualization of biomolecules using their molecular mass, and can be used to classify tumors, produce molecular images with substantial spatial resolution, and collect significant biochemical information. We then took the results of the glycogen and glycan abundances from each technique to analyze and further understand the role of glycogen and glycosylation in the progression of brain cancer. The first results we collected were pertaining to glycogen and taken from both the IHC and MSI platforms. As you can see in figures 2A and 2C, there is a direct relationship between glycogen abundancy and brain cancer grade level. This is also visually represented in figure 2B with the halo images and in figure 2D with the MSI images, where cooler colors correspond to lower amounts of glycogen and hotter colors correspond to higher amounts of glycogen. We then collected more results pertaining to glycosylation. These results were taken solely from the MSI platform. And as you can see in figure 2E, there is a connection between glycan abundancy and therefore glycosylation and brain cancer grade level. For some glycans, as abundancy increases, brain cancer grade level also increases, similarly as to the glycogen. However, for other glycans, as abundancy decreases, brain cancer grade level increases. This is true for the select glycan 1257 that is also visually represented in figure 2F. You can see that the control image is dominated by mostly hot colors, representing a high glycan abundancy. And then, as brain cancer grade level increases throughout the images, they become dominated by cool colors, showing a decrease in that abundancy. Overall, our results are important because they reveal significant insight into the composition of brain tumors. We hope to conduct further research into related mechanisms, and excitingly, the MSI platform has recently been utilized in the clinic for lung cancer to define the edges of cancer tissue and guide resections. We hope that our work may also be utilized to guide these critical decisions in the clinic. And with further research, our work may lead to the discovery of biomarkers and novel targets for the diagnosis and treatment of metastasizing brain tumors in humans. If the signs of brain cancer are more extensively recognized, an earlier interference in the progression of the disease can occur. And this advancement is pertinent to the standard of living of the estimated over 700,000 people living in the United States today with a brain tumor. Thank you for listening to my presentation, and I'm happy to take any questions.
Thank you very much.
Thank you.
Well done. Kayli, is that how you pronounce it?
Yes, Kayli.
Excellent, thank you. I got a couple of questions for you, I really do. So first, you said about 87,000 per year.
Are diagnosed with a brain tumor.
Is that in the United States or globally?
Globally.
So that's a global number, okay. Not terribly reassuring either, but you know. And again, this goes under, as many things tonight, my ignorance. But the presence of glycans in certain cases, or the decrease in the glycans, increase in the severity of the cancer. Glycogen is the opposite. So can you just clarify for me again the difference between glycogen and glycans, because it's all sugary?
Yes.
Right?
Yes, these are both sugars, and they're both very important for our bodily function. With glycogen, it's going to be the carrier of glucose. As we talked about earlier in some of the other presentations, glucose is a complex carbohydrate that is very important for our metabolism, specifically in glycosylation. The glycogen in our metabolic pathways. And then glycans are important because they're also sugars. However, they are more important for in the cells. They can attach to things like proteins and affect how those proteins function and their structures. So both sugars and very important in metabolism, but a little bit different in the functions.
Great, thank you. And then I've got sort of very theoretical. I really appreciated the fact that you talked about what the potential applications and implications of this is, and in the importance of early diagnosis. But it seems to me one of the challenges with these sorts of cancers is that you have no reason to go in and do scanning or do tests until it's fairly far along. Again, beyond what you said here, I'm just curious your thoughts on how there gets to be a better awareness of when to go ahead and make these sorts of tests. What is it that we can do to help, if we're going to have 87,000, how do we get to them early enough to have more therapeutic treatment?
Yes, I believe that right now this is a very invasive test, and not everybody is going to be getting their brain tissue taken for testing. So right now that is a limitation, but with further research, we're hoping that if we can find other biomarkers that we can look at and find these similar connections, that we can use those instead to potentially find high-risk patients. One example could be cerebral spinal fluid, which is still invasive, but much less invasive than taking brain tissue. Thank you for your questions.
So what led you to believe that glycogen and glycans would have a role in revealing the extent of the malignancy or the extent of the cancer?
Thank you. In our lab, we study a lot of different glycogen storage diseases. And so when we are doing metabolomics and looking at potentially very important metabolites in different kinds of samples, there are many diseases that we're looking at, such as Lafora disease, different types of cancers like brain cancer, Ewing sarcoma. So it is a theme that we go in with our lab. We're looking at different diseases that we can target glycogen, and it's going to be important. So it's what we're looking for kind of going into it with multiple different diseases.
With respect to, let's use glycogen as an example, why do we see the increased prevalence of glycogen as you increase in the grades of the cancer?
I do believe that that is a little out of the scope of this particular study, but that is something we're definitely thinking about. And right now we're seeing that this is a connection, that we are seeing these patterns, but in order to really pin down why it's happening, that's where we'd have to do the further research into related like metabolic pathways and those kinds of things. Thank you for your questions.
So Just let me ask a question for clarification. These are actual physical samples of human brains that you're sectioning here and then examining with different types of instrumentation.
Yes, ma'am.
And so I think you said before that not everyone is going to have that kind of intervention, so to speak, until maybe quite late in the development of their disease. Is there a possibility then that you see down the road that glycogen could be examined through some non-invasive technique, or is this the kind of really a cell-based study that requires the physical sample directly on the slide under the microscope?
I do believe that it will need to be somewhat invasive. Like I was saying, we may be able to find biomarkers that are less invasive, such as the cerebral spinal fluid. But as of right now, it is still very invasive. And there are ways that this can also translate into a surgical kind of aspect, like I was talking with the lung cancer. So if somebody is getting a tumor removed, something like this would be good because a mass spectrometry knife could be used, where as the surgeon is cutting out the tumor, they can see in real time where the edges of the tumor are, and it can guide them. So it can not only be a tool in diagnostics, but also in treating the cancer or removing the tumor.
That's fascinating. Thank you very much.
Thank you.
Does a mass spectrometry knife exist?
Yes, it does. Yes. It's not being used for brain tumors, but it's very excitingly being used for lung cancer in the clinic. Thank you.